sorry, that last video got cut off, so I might repeat a little bit here. But once we cut up the DNA with restriction enzymes, we cut it into smaller pieces so that I can see the multiple parts of it. I place it into the gel that you can see here, and we've dyed the DNA, which is why, so that we can see it. And so we see the strands here. And again, when we turn the power on, because of the opposites attract and the similar positive negative repel each other, then we can push it through, and it's kind of like pushing it through the sponge. So we know that the gel is a little bit porous, even though it looks kind of solid, it's not. It's got pores in it, similar to this sponge. So the restriction enzymes cut it up into different size pieces. So obviously the larger ones have a much harder time moving through the gel, so they can't move as fast. And so the smaller ones can squeeze through all these pores relatively quickly and with ease. And so we see if we're going from this direction that you can see my mouse moving from top to bottom, down here are going to be my smaller pieces because they've moved further in the same amount of time. And up here I'm going to generally have my larger pieces. And so when I read the gel, I can compare them. And these strands, if they're from the same person, the enzyme should have cut them up into the same pieces. And so all of these things should match each other. So here you can see I have this big plastic thing here is what I put it in. So I have my power source. This is what the gel sits in. And these are my little wells cut out. And so I will put my DNA samples into these wells. I turn the power on and it pushes, the electricity pushes it through. So DNA being slightly negative is going to be repelled by this charge, is going to be attracted to this end, and so it's going to move through the gel this way to create the bands that I see. So when do I use gel electrophoresis? Again, we use it for DNA fingerprinting, but it's used for paternity cases, so who's the daddy? It's used in forensics, so crime, so basically who did it. And then we can also use it to see every evolutionary relationships. So basically looking again, the more DNA sequences I have in common, the more closely related I am. So if I put a sample in, like if I have a giraffe here and then I put in 2 or 3 other samples, the animals that it's most closely related to are going to have the most number of matching bands. And so we'll do an example in a second. But because everyone's DNA is unique, that's why I can use it in terms of paternity and for forensics to match up basically who did it or who's the daddy. Okay, so here's an example. So whose DNA was left at the crime scene? So basically this is a drawing of what I would get. So all of these lines would essentially represent my DNA pieces. So this is the DNA that was left at the crime scene. So I want to know if it was Bugsy, Dino, Daisy, or Tootsie that did it. And again, I have to have exact matches here. So if I go across here, my only possibilities here, this one immediately eliminates Daisy. So it could be Bugsy, Dino, or Tootsie. Then I go here. It could be any of those, but again, I've already eliminated Daisy here. Bugsy and Tootsie are now out because I am missing a DNA piece here while they both have it. Again, so I've already eliminated Tootsie, eliminated Daisy, eliminated Bugsy. So by process of elimination, it has to be Dino, but if I go in, I can match all 4 spots. Dino doesn't have any more. He doesn't have any less. And so Dino is the one who did the crime. Who left the blood stain? We can look at this one, but we're going to practice with this in class as well. But again, I have to have a complete match here. So just looking at it, if I look for this one right here, the only person who has that in here as a double section at this marker right here would be John. But if I look at all of them, John has the matching correlation to all of it. He doesn't have anything extra. He doesn't have anything left over. Evolutionary relationships. If I want to know which organism is species A most closely related to in terms of X, Y, and Z, I have to go and count how many they have in common. So species A has all of these in common. With if I look at this marker here, I know that now this gives me 2 commonalities with species X, where Y and Z still only have this one. Right here gives me species Z another one, so X and Z now have 2 commonalities. Right here gives me one for species Y, so they all have 2 commonalities, and so this is going to be my tiebreaker, my determining factor. And because species Z has the same marker, we know that A is most closely related to species Z. So as we find fossils and everything, we can extract DNA from the bones, the fossilized remains, and we can compare it to organisms that are alive today, and we can tell evolutionary relationships, and we can kind of build those cladograms to see how species changed and evolved over time. Which kids are dad's biological kids? So we know that so if you were D1 right here, kids get half of their DNA from mom, half from dad. So as we look at mom and dad, you're not getting all of mom's traits because some of these you might have gotten from dad, and again, you're not getting all of dad's traits. So each kid is going to kind of be a mix-up of mom and dad together. But the child can't have a marker at a point where mom and dad don't have a marker. So basically, if we look at D2 right here, we have some red strands, and if we kind of follow this across, we know that this didn't come from dad, definitely didn't come from mom, and so D2 can't be either one of their kids. And again, if I look at this red band right here, it definitely didn't come, obviously it's color coded here, but it's a little bit higher, it's a little bit hard to see, but it's supposed to be kind of like in the middle of this blue and this yellow here, and we know that it definitely didn't come from dad, definitely didn't come from mom. So when you're looking at the child's DNA, if you're given mom and dad's DNA, you know it's a mashup of both of them. So if you look at daughter 1 and son 1, you can see they got this one definitely from mom, they got this one from dad, this one from dad, and this one came from mom. So child has to have a marker where their markers are. One of the two parents has to have it. It doesn't have to be both, but only one, because you get 50% of your DNA from mom, 50% from dad. Same thing with son 1. We can see he got this marker from dad right here, this marker from mom right here. These two came from dad, and then this last one came from mom. Okay. Again, paternity case number 1. Here's the mother's DNA, right? So they definitely didn't get DNA strand number 3. So could the suspect be the father of this child? Well, we know 1 came from mom, so 2 had to come from dad. Does dad have a marker at this position? No. So this suspect number 1 could not be the father. Okay. And so that's what this is going through. Child inherited band number 1 from mom. Child inherited band number 2 from biological father, and it sure as heck isn't this guy. So we got to keep looking, Maury. All right, so band number 1 came from mom. Could this person potentially be the father? Yes, absolutely, because band 1 we know came from mom. Band 2 could have definitely come from this person, so we know that this guy, I would say, yes, is the biological father. Okay, and so that's how we do DNA fingerprinting there. Again, we use it for paternity, we use it for court case suspects and crime scenes, and then also evolutionary relationships. All right, the last part of this, gene recombination. This is my actually genetic engineering. So if I take genes from 2 different organisms and I put them together, then we have a recombinant DNA because I have pieces of DNA from 2 or more different organisms. So basically what happens is I cut the DNA with restriction enzymes at certain points to take out certain genes that I want, and so that's called CRISPR technology. You may have heard it mentioned in class, but we can basically design these enzymes to find any sequence of DNA that we want. So the Human Genome Project mapped out all 46 chromosomes and what the bases and sequences should be. So we know exactly what should be there, what proteins are made, what the sequences is for certain proteins. And so we can create an enzyme, a certain restriction enzyme, if we know what the sequence is. We can basically program the enzyme to cut out a certain gene segment for us. So the first step is that we have to cut the DNA with restriction enzymes. Then basically we take that new gene and we glue it into place into a different organism. And so when transcription and translation occurs, the new protein or gene will then be made in the organism that we put it in. So kind of like the same thing that was happening with our insulin there. So organisms that have these new genes or new traits, we call them transgenic organisms because they have multiple genes from different organisms, so it's not necessarily from them. So again, we have to remember that the DNA code is the exact same for all organisms. So the machinery, the mRNA, the RNA polymerase, all of that stuff is going to work exactly the same in all organisms. So they don't know where this DNA came from. They just read it as if it's normal and it's supposed to be there. So a lot of times where we use this technology for transgenic organisms is in GMOs, or genetically modified organisms. And so we use this in agriculture a whole lot. And so what we see is that we're producing crops that are resistant to drought, or they're resistant to certain pesticides. So if we know that there's a gene of a certain plant that can live in dry areas, it doesn't need a whole lot of water, we can use a restriction enzyme, cut that gene out, and insert it into a crop that generally would need a lot of water, and so then it can survive in high drought areas. So this is beneficial for farmers because it's going to produce, their crops aren't going to die, so they're going to produce more, they're going to get more money. But on the flip side, they're also going to spend less money in having to water their plants. So it's kind of a win-win. But then we hear all this crazy stuff about GMOs in the news and on the media, and so people are really freaked out about it, and that we're genetically modifying organisms, but it's not necessarily, there's been nothing linked to GMOs and that they cause certain diseases or things like that. There's nothing like that that's ever been proven for genetically modified organisms. So even though the technology is there, it's not like we're creating all of these crazy mashups of all of these different organisms. Mind you, a pig with a glowing nose is a little bit extreme, but it just shows that the technology is there and available that allows us to do this stuff. Okay. So another example is a transgenic mouse. So the gene for feeling full was removed, and so this mouse just eats and eats and eats and eats because it never feels full. It doesn't have the gene, and yes, that is a genetic disposition to feel full. So when you take it out, this mouse obviously just kept eating, got obese, and so we've used this in a clinical study to study obesity to hopefully help people that have issues with obesity. Goats can spin webs, so the gene for silk production is put into female goats. So their milk contains silk, and silk is a very strong fabric, and so we milk the goats and we get silk out of it. So obviously he's not spinning a web out of his head. It's through their milk, and so we can collect that silk and then we can spin it and create fabric from it. Disadvantage, for some reason, and we haven't determined why yet, but the goats usually die a little bit younger, and so obviously there is a lot left to be determined in terms of the safety risks of this, and we also, we don't know why these things are happening, so obviously these aren't great methods or mechanisms to be using for all sorts of things yet. There are lots of ethical issues in terms of that. We've talked about this, crops resistant to droughts. They can still grow in small amounts of rain. It's really great for the economy. Is there a long-term effect on human health? I don't know. We haven't been using it for that long, maybe 10 or 15 years, if that. So I don't know, 50, 60 years down the road, we might determine that there are health effects, but as of right now in the short term, in terms of maybe the last 10 years, there hasn't been anything noted in terms of health risks. Okay. So look this over. This is also printed out on your sheet. So we have benefits of GMOs. We also have risks of GMOs here. The biggest risk here is unknown effects. Okay. We also have to think, if we're growing crops out here, we can't localize the pollen to just that crop. So we also don't know, as the pollen blows in the wind, and it's cross-pollinating with other ears of corn, or if we're growing flowers and they cross-pollinate with wild flowers, what are the effects that we're creating? So some of these cross-pollination things could lead to super weeds. Instead of superbugs and bacteria that are resistant to antibiotics, we then get these weeds that could be really harmful to the environment, and we just can't kill them because we have introduced some of these new genetic traits to them. Cloning. We can go over this in class, and there is a mouse simulation here, but basically cloning is not done with all embryologic cells. I'm going to show you how that does it in this next video segment.